With our experiment, which is determination of the solution of solid dosage form, we will make the solution profile and withdraw it for paracetamol tablet 500 milligram using the solution apparatus 2, which is paddle type. And we have the solution apparatus 1, which is basket type. The required dissolution apparatus is determined by USP. So, by opening USP on the monograph for paracetamol tablet, we can stabilize, or we can use the paddle type, as we said before, 900 ml of phosphate buffer with the pH 5.8, adjusted the temperature to 37 Celsius, and the paddle will rotate 50 rounds per minute. The time of our experiment is just 30 minutes. It's quite enough. We will withdraw 6 samples at each 5 minutes. So we will use syringes to withdraw our sample. So our sample will be each time 5 ml using a syringe. The sample is withdrawn from the midpoint, not from the top, not from the bottom. So after withdrawing the 5 minutes, it will be filtered using syringe filter and collected in a small bottle. In order to get accurate results, we need to do dilution using the same phosphate buffer. So the dilution factor will be 50 times. So we withdraw 1 ml added to 50 ml and completed with the phosphate buffer to 50 ml. Then it will be mixed up and down, and the absorbance is measured using at lambda max 243 nanometer. This is the first sample. After I draw the first sample, I have to adjust the volume of the dissolution media to 900. So I need to do replacement by adding 5 ml of phosphate buffer each time. I will make this step 5 times because the last step will need no replacement. Okay, this is the dissolution apparatus, paddle type 2. It is adjusted to 37 Celsius, and the paddle will rotate 50 rounds per minute. I'm using Rivaline tablet, which is paracetamol tablet 500 milligram. I will add just 1 tablet, and by measuring the time as 30 minutes, as we said, because each time interval, which is 5 minutes, we will withdraw 5 ml from the dissolution media. So we will start by making it rotate. It's adjusted to 37. I will add my tablet and measure the time meanwhile. Five minutes is finished. So after the 5 minutes is resumed, I will withdraw my sample from the midpoint, as we said before, not the bottom, not the top. I withdraw 5 ml using the syringe and adjust the volume. So this is my sample. So to make the volume of the dissolution media is stable for the next sample, I will add the same dissolution media, which is phosphate buffer 5 ml, and I will resume the experiment. This is my sample. I will use a filter, disc filter, to filter my sample just by adding it like that and collecting in a small bottle. these have to be discarded. This is my filtered sample. I have to dilute it 50 times using volumetric pipette 1 ml, and I will add it to volumetric flask which contain a lot of phosphate buffer. Okay, so just by withdrawing will be diluted 50 times, then I will complete the volume to 50 using the phosphate buffer, the cold phosphate buffer. My sample is diluted in order to enable me to measure it using UV and get a value between 0.2 and 0.9 as the absorbance value. I have to see the meniscus, then close it, invert it. This is my diluted sample. Last step is to measure the absorbance using UV at lambda max 243 nanometer. This is my cell. I will add the sample, and UV is adjusted using the phosphate buffer as a blank. I will add my sample and measure the absorbance value.